So welcome, everyone. Thanks very much for coming, especially at this relatively early hour for most of us academics. My name is Nathan Urban, and I'm the head of the Biological Sciences Department here at Carnegie Mellon University. And with Mike Tarr and some others, Greg Travath, Tyler Galson from Butterfly Networks, we've been the organizers of this meeting, this workshop. It's my great pleasure to introduce Jonathan Rothberg, who's the sponsor and really the original motivating force behind putting this meeting together. Jonathan is an alumnus of Carnegie Mellon University, I believe 1985, from the CIT, a chemical engineer, who then went on to do his Ph.D. At Yale. Since then, Jonathan's career has really been characterized by one set of technical innovations and practical applications of those innovations after another, especially in the area of gene sequencing, although his interests are clearly much broader than just that. Most recently, he went through and developed really one of the most interesting and exciting technologies for next generation sequencing, the Ion Torrent device, which the Ion Torrent company, which was recently sold to Life Technologies. And now it seems like yet another round of interests and explorations is something that Jonathan's begun. And in part, those interests in that exploration is what's resulted in this meeting today. So thanks to everyone for coming. I really appreciate you being here. I'm looking forward to a very, very exciting meeting. And with that, Jonathan is going to kick us off.
Thanks. We'll see how the sound gets picked up. Thanks for everybody coming this morning. When I give lectures, and I do talk about DNA sequencing, most of my talks start with the birth of my eldest son. He wasn't breathing, and I wanted to understand his genome. And that led me to develop what became the first high-speed DNA sequencing instruments and next-gen sequencing. And when I introduce sequencing, I like putting it in historical context. I believe that DNA sequencing will be one of the foremost important advances in medicine over the last century. So I like saying things like clean water. That was probably the most important. Antibiotics. That might rank up there as number 2. And I throw in imaging. Say, that was number 3. And now we have DNA sequencing. So after doing that for a while, I figured I better go back and do some homework. And I realized I didn't know much about imaging. And I thought that I should learn more. And I told a bunch of guys here who are Butterfly that the best way to learn things is to bring the smartest guys in the world all together in one room and listen to them. And so that was really the genesis of this. Maybe a little bias from the sequencing, next generation medical imaging. I start with this slide, one, because I got to introduce my son, who recovered, and got to introduce him to Gordon Moore, and we actually sequenced Gordon Moore's genome. But I actually think this is an important slide, because most of the things we do, we probably take for granted that over the last 40 years, there has been a million-fold improvement in computing. And I know, as I said, in sequencing, this has had a dramatic effect on our abilities, both leveraging this technology directly to sequence, as well as leveraging these advances to understand that sequence. And I thought one of the macro themes for this conference would be to rethink the last 40 years of imaging, whether it's ultrasound, CAT scan, or MRI, and rethink it in context of what we can do with 1 million times more computing power. So that was, again, the impetus behind this. And one of my favorite comparisons, and I can date the people in a room by how many people recognize what's on the left here. And I remember when I was an undergraduate at Carnegie Mellon going up to Westinghouse and sitting on that couch. And as many of you know, that's not a couch. That was the world's most powerful computer. But what was really amazing when I was looking at a chart of Moore's Law was that the new iPad actually is 20 times more powerful than the Cray 1. And for those of you who remember, that couch on the left is 25 million bucks. The tablet on the right is about $800. So Moore's Law really has been fantastic. And as we'll discuss today, I hope that same power that it's brought to your computing devices can be brought successfully to imaging. A little bit on next-gen sequencing, just to show you some things. I like showing these slides because a lot of people, when they talk about DNA sequencing, and I hear this over and over again, and I've actually never heard a rebuttal to it, I hear over and over again, it's moving faster than Moore's Law. And I think it's just an illusion. And in fact, it's just catching up to 40 years of accumulated Moore's Law. And I can give you some specific cases. So I was lucky enough to be involved in the first DNA sequencing high-speed machines at a company called 454, and we made great progress in terms of the billion of bases of DNA that we can do in a day, and we were able to go from sequencing viruses to bacteria. Just to show the power of it, we actually sequenced Neanderthal. You can sequence somebody who hasn't been around for 40,000 years. And then to show the promise in individual medicine. So many of you know about the Genome Project, but the Genome Project wasn't about an individual. It was about 50 people being mixed together and understanding what it meant to be human. So this was the rate of progress of the first DNA sequencing technologies. This was a fast follower. Sometimes I tell people this is when they fired me. This was a fast follower. And then we came back into the business. And what we did is said, let's not leverage Moore's Law indirectly on informatics, or conceptually, like using monolithic idea, let's just use semiconductors. And this is Ion Torrent, actually, semiconductor sequencing. And actually, this month we're shipping a machine that can do a human genome for about $1,000. And so Moore's Law has been really great. And I just use it as an example of catching up to Moore's Law. Now, today you'll hear, and tomorrow you'll hear a number of talks on imaging. But I did just take 5 minutes to Wikipedia the three things that I always talk about. I always tell people imaging, and I say ultrasound, I say CAT scans, and I say MRIs. And you really see that these guys did amazing things given what computing looked like in these respective years. Really amazing things. And people will tell me, that's because they were smarter back then and had smarter algorithms. But I do think it's an amazing opportunity all of us have to revisit these modalities with this millionfold more computing power. Before I turn it over to everybody, I did want to give some thanks to Nathan and Michael from CMU. And I am giving a plug. I do trade in a currency. The currency I trade in is smart students. And my email is jonathan.rothberg@gmail.com. And I'm asked to give talks, and I tell people I'm happy to give talks if you send me students. And so here are three bright guys that are working at a new startup, and their mission is really to apply Moore's Law to medical imaging and take advantage of these 40 years of accumulated Moore's Law and see how they can do things better, faster, cheaper, to democratize these technologies and allow them to be used around the world. So I want to thank not only the CMU guys, but the guys from Butterfly. And the Butterfly guys are always looking for smart students. So I will say that a couple times. Okay, so I want to make sure everybody enjoys the next couple days, and thanks for coming.
Our keynote speaker, first speaker of the morning, is Dr. Jeff Lichtman from Harvard. Jeff's going to talk about some approaches which really generate some of the largest data sets that I'm aware of in the imaging context, and what these data sets can be used for And really the long-term goal in this area is not genomics, but connectomics, trying to understand and map all of the connections in the given brain areas. And Jeff is doing remarkable service to us because he's actually going to be also in Pittsburgh this weekend, but he has to go back and teach in between. So he's making quite we really appreciate his efforts.
Well, I'm extremely grateful to be here and have an opportunity to talk about imaging. As you've mentioned, I'm teaching an undergraduate course, 250 students yesterday and another 250 tomorrow. So I am a little squeezed for time, and I'm sorry that I won't be able to participate in this symposium for very long. Medical imaging is, of course, all about taking advantage, leveraging our visual systems to apprehend some disordered aspect of human organ systems. More often than not, I think we are aiming to reveal a structural abnormality in an organ. And I think with one notable exception, humans have been enormously successful in this aim. From the time humans could dissect cadavers, a pathologist doing a gross dissection could often, with his naked or her naked eye, ascertain problems in many organ systems just by looking, just by seeing a liver that's diseased. With your naked eye, you can usually tell what's wrong with it. Of course, with the invention of microscopes, it was possible to see changes in the cellular architecture of organs. And I would argue that, again, with one notable exception, the use of light microscopes and special stains, virtually every disease of every organ system, with one notable exception, I keep saying, can now be seen and put into our nervous systems through our visual system, which is our strongest organ for understanding the world. In a few instances, when the defect is too small to see with the light microscope, people push on to the higher resolution of the electron microscope, and that's brought to bear. But we have done really a great job of physicalizing disease in the sense that we have in our mind now a mental physical structure of the abnormality for all diseases and all organ systems except for one notable exception. And the notable exception is the nervous system, where there remain a large number of diseases. There's nothing up there. It's just me at the moment. I know this is so weird. You have to pay attention to me. There's a large number of diseases, common diseases, that anyone can see from the outward manifestations of the disease that there are things wrong with a child with a severe learning disorder or behavioral disorder or an adult with some form of mental illness, that there's no question something is wrong. But when you try to find the physical embodiment of these abnormalities, we fail dramatically. And this is an important problem. Why is it? Compared to all the other organ systems, the nervous system has remained so complicated. So here, compared to other organ systems, the relationship between the structure and the function of the nervous system is much more complicated. And I'd like to explore a little bit why that is. In part, this is a way of rationalizing why people might want to take the extraordinary steps that are necessary in order to understand how the nervous system works. So there are many different reasons for this, but one thing that is clear is that if you take a light microscope and look at a liver, and it can be the liver of a frog or the liver of a human being or any animal in between, you see a cellular motif that is recognizable and that gives you an understanding of the basic function of the liver. The same with the kidney of any animal that has a kidney. Although the kidneys are different shapes in general, when you look inside, there is a cellular motif that is common from one animal to another. And not only that, that cellular motif is iterated many, many times in the organ. So once you understand that cellular motif, for example, in a kidney, the tubule system, once you understand one such tubule, you basically understand the whole kidney, because the kidney is just made up of hundreds of thousands or even millions of tubules like that. The alveolus in the lung is another example. The acinus in secretory glands is another example. What about the cellular motif in the nervous system? Well, the problem is that the nervous system is just vastly more structurally complicated than any other organ. For example, there is, if I asked you how many kinds of cells are there in a liver, different kinds of cells, you might come up with a number like 10 different cell types. In the nervous system, this number of cell types is not known. In fact, a state-of-the-art question is how many different kinds of cells there are in the retina, which is the most well-organized, most well-behaving piece of nervous system. It's laminar, everything is tiled in a geometric way. It is a state-of-the-art question, and Moritz Helmstetter is one of the experts in this who can attest to this, to just decide how many kinds of cells are there. How many kinds of cells are there in the retina? More than 40. Okay, and that could be 400 or 100. I mean, it's amazing. We don't even, no other organ would this still be a question. And eventually we will have an answer. Do you agree? And that'll be great for the retina, but it's not going to help us with how many cell types there are in the cortex, or the cerebellum, or the brainstem, or the basal ganglia, or the spinal cord. Every part of the nervous system has this extraordinary number of cell types. This is just explosion of diversity that you don't see in other organs, and that diversity also gives rise to an extraordinary diversity of function. Whereas you could write a chapter, maybe 100 pages long, that describes everything a kidney does, how big a book would you have to write to say everything that a human brain does? Could you write a book like that? Of course you couldn't, because every day, every week, people invent new tools, people are using their nervous systems in new ways, and this means we have a different problem of diversity in function, in that the functions are widely ranging and changing over time. So that's one reason it's harder to get at the structural underpinnings of abnormality, because the structure of normalcy is not even known for the nervous system. That's not the only problem. The second problem is that whereas in the kidney, as I mentioned, once you understand one little piece of it, you basically understand the whole thing. Once you understand one little piece of lung, you basically understand the whole system. Not the same in the nervous system, where there is organizational principles at many different size levels. And this is a picture that is from a review that Winfried Denk and I published last year, just showing the human brain in cross-section at 10 centimeters all the way down to a little box at 1 micron. And I'm just putting this up here to remind you, if you don't know this, that at this level there is extraordinary division of labor. The occipital lobe in the back of the brain is important for vision, the cerebellum for balance, breathing and stuff in the brainstem, the spinal cord for movement. Just at the centimeter level, one finds a lot of important information, and this is why the imaging field of functional magnetic resonance imaging, using the blood oxygen level detection technique, for example, which gives resolution of cubic millimeters, is very valuable in the brain. Although that might seem a very coarse way of looking at the brain, it allows one to parcellate out the different functions of different parts of the brain in normal and diseased brains. If one carves up a little piece of brain and looks in detail at the cerebral cortex, one finds the nerve cells are at the top, and then there are these long areas that are called white matter, filled with cables that connect one part of the brain to another at the scale of centimeters. One sees that. If one looks at one little piece of the gray matter, one finds that it's filled with nerve cells, which, unlike cells in any other organ, are insanely complicated. This is a nerve cell body of a pyramidal neuron, a kind of neuron that populates the cortex. It has a bunch of basal dendrites that are receptive for this cell, and an apical dendrite that comes up very close to the very surface of the brain, and this is also receptive. And in addition to these receiving processes, each of these cells has a single axon that can span, cross over to the other hemisphere, go down the spinal cord. That is the output of this cell. every single brain cell is itself divided into these different areas, and each of these areas is taking information in from other cells. If we zoom in on a little part of one of these cells, let's say the apical dendrite, one sees that the dendrites themselves have little spines coming out of them, and that's where the synaptic connections from other cells are made with this cell. Whereas the shafts, the areas that don't have spines, is where many of the inhibitory inputs to this cell are. So there's a huge amount of diversity of what's going on, even at the level of little parts of this cell. If one wants to get even more picky, one can zoom in on one of these dendritic spines, and here in cross-section in the electron microscope, you have to go to this kind of resolution to see what's in the spine. There's an apparatus down there, and in the synapse of an axon that's making connection with that spine, there are a bunch of synaptic vesicles. Their number, their placement, the localization of the postsynaptic density, a huge amount of details about the fine structure of the way cells communicate occur at this level. There is important information everywhere from this level to this level. That's 6 orders of magnitude. And just to make this point a little clearer, the resolution of an image up here might be a cubic millimeter. The resolution of an image down here might be 100 cubic nanometers. An fMRI voxel, a cubic millimeter, is 1 trillion times larger than an electron microscopy voxel. And yet if you want to understand the brain, you have to know things all the way from this level to that level. And trillion is still a big number. No matter how you cut it, that's a big number. And that's just the levels of resolution you have to overcome to understand the brain. But those are the easy problems. There's even a worse case. And the more serious problem, especially with human nervous systems, is most easily summed up by thinking of this simple fact that a lot of organs, including the brain, get their information from genetic instructions that generate a structure that ultimately generate the function. That's true for every organ of the body, including the brain. But that's not the end of the story. And with humans, it's not even the big part of the story. And I'll give you a good example. Has anyone here not learned to ride a bicycle when they were a child? Is there anyone here, you never rode a bicycle at all as a child? Do you ride a bicycle now? How did you find learning? When did you learn?
15, 16.
Oh yeah, you're still pretty young. How hard was it? It wasn't that hard?
It was hard enough.
I have a neighbor on my street, a woman who was born and lived in Africa as a kid. She moved to Cambridge. She has 2 little kids, and last summer, her kids are riding bicycles. One's 5, one's 7, or around that age, and she decided she wanted to ride with them, and we're on a pretty quiet street. So as an adult, she got a bicycle. And as far as I can tell, she's a neurologically normal adult woman in all respects, except when she gets on a bicycle, where it looks like she doesn't have a cerebellum. And she's just going back and forth, falling over, and her kids are riding circles around her, literally, saying, "Mommy, what's wrong with you?" And she's getting madder and madder. And she spent the whole summer mastering a bicycle. I should put mastering in quotes, because she never mastered it, and she's not been on a bicycle again. I think, and an adult trying to learn to ski often finds a similar problem. They see these 5-year-olds going down the slopes, and as an adult, they can't. So I learned to ride a bicycle when I was a kid, and then I moved to St. Louis, where everything was done by car, and for 30 years, I did not have a bicycle. And then we moved back to Cambridge, and my wife noticed about 7 years ago that my belly was getting larger, and she suggested that I take up exercise, and why not bicycling? So we got bicycles, and I got on a bicycle after 30 years of not riding a bicycle. And it was 8 seconds before I was perfectly fine. That's after 30 years of no practice. 30 years, and it took 8 seconds to feel comfortable. And I look at this woman who spent hours every day and couldn't, and I would say, well, what is the difference? Do I have a gene for bicycle riding that she doesn't have? Of course not. Something about using the nervous system, practicing, balancing as a child, has changed me in a way that has lasted 30 years and not required further input. So there is this loop. And this loop is a horrible thing because biologists hate loops. Loops mean there's no cause and effect. It just keeps running around here. We love this. This is why there's such power in genetic work, because you start with a genetic change that changes the structure. It's a linear path. But when you get into loops, it gets more complicated. Of course, genetic information itself there's huge feedback loops in the dynamics of when genes turn on, which is still deeply mysterious. We don't have good maps of the dynamics of gene expression, but we certainly know that genes cause this and this causes that. Cause, effect, cause, effect. But once you have loops, there's no longer a cause and an effect. You learn something, it changes the way you do something, and the way you do something changes the way you are, and that keeps going. So this requires, if one wants to get at this, one really has to deal with what kinds of structures would instantiate riding your bicycle. Is it going to be, there's a gigantic pathway in my brain that doesn't exist in the brain of my neighbor? Not likely. It's more likely at the level of the wiring of the actual connections. There are some subtle differences between me and my neighbor that allow me to ride a bicycle and she not. And that's not a trivial and just a weird thing. It's not just bicycle riding. It's buttoning your shirt in the morning. It's reading. It's using the language you use to think. Everything we do as human beings, pretty much, is coming through the turning of experience into a physical embodiment that is indelible and lasts a lifetime. These embodiments of memory used to be called engrams, so this process of turning function into structure might be called engrammatization. I made up that word, of course, so it's not really a word, but that is the idea of what I'm personally deeply interested in. What would engrams look like? And to see them, one has to image, and to image, one has to map out a wide amount of complexity. And this is a mapping problem like genomics is, and so therefore one steals this term. Instead of mapping genes, one's mapping connections, so it would be connectomics. Now that's a new term for most of you, so I've brought the dictionary definition here. Connectomics or connectomics. It's a plural noun, but singular in construction. This, by the way, is from Merriam-Webster's Unabridged Dictionary in the year 2015, or not. When I first wrote this definition, it was 2013, and I realized I'm still pushing this word out there. But this word's being used a lot, and so I have a feeling that the omics revolution is going to come to this deep question of how the brain is organized. It's a branch of biotechnology. It's important to realize this is not biology, it's biotechnology, concerned with applying the techniques of computer-assisted image acquisition and analysis to the structural mapping of sets of neural circuits or to the complete nervous system of selected organisms using high-speed methods. Just like in genomics, speed is everything. With organizing the results in databases and with applications of the data, as in neurology and psychiatry, there almost certainly are pathologies of connections, connectopathies, if you will, where unless we have ways of seeing the wiring, we're not going to understand what's the proximate cause for abnormalities in human behavior. And there are fundamental neuroscience questions, such as the one I just mentioned about engrammatization, where this would be very valuable. And I just stole the definition of proteomics and genomics to make this sound this way. This is basically the same language, just attached to connections. And in this dictionary, one might also find the connectome, which would be like the genome, the full wiring diagram of an animal. In my lab, we've been working on two different approaches. One, an optical approach using light microscopes, and the other, an electron microscopical approach. With the light microscopical approach, we have found evidence for a certain number of cellular motifs associated with experience. I'm going to briefly tell you about that. And then a much harder question is using the electron microscope to find cellular motifs in the nervous system, perhaps associated with experience. But that's where my own interest is. So for the light microscope, we've taken advantage of the fact that there are many colors of fluorescent proteins, of proteins that are genetically encodable through the genes for those proteins, And this comes about from the remarkable discovery originally by Shimomura and then helped by Roger Chen and Marty Chalfie in turning fluorescent proteins into a tool that would allow cells in animals to be colored, even though these originally came from marine animals, the jellyfish to begin, and then corals. Now, and mutations of these fluorescent proteins have given us a wide range of colors that could be used to colorize the nervous system. And when we began thinking about this, and by we I mean Josh Sains and me, we were thinking, how many colors does one really need to understand the nervous system? And it turns out, of course, that if you have a gene for a green fluorescent protein, a red fluorescent protein, and a blue fluorescent protein, you basically have enough colors to populate all the color space a trichromic visual animal like a human being has. This is, of course, how black and white went to color television when there were 3 phosphors, 1 for each color. So if you mix different amounts of red, green, and blue fluorescent protein in individual nerve cells, you could, in principle, have a brain where every single nerve cell has its own color, or at least as many colors as you could get out of a color television or a computer monitor. And so we built, through the work of Jean Levé, a postdoc of mine, some clever, it's the cleverness of Jean Levé, genetic constructs that we could put into mice that would allow, under control of a promoter that expresses these constructs in the nervous system, individual cells at first to be one of 3 colors, which is a far cry from millions of colors, but that's where we started. And that was to make a construct that had 3 fluorescent protein genes in it, and then through the use of an enzyme, we could make this construct either express the first color, which it did in the default state, because there's a stop signal, so it reads from here the cherry color and then it stops. But then in the presence of this enzyme, this could be cleaved out, in which case this would cause the second color to be expressed. But there's a choice, and sometimes both are cut out, in which case this promoter only allows the third color to be expressed. So that's what we started with. And this is a brain of an animal where the astrocytes, the glial cells, are one of those 3 colors. I'll just zoom in on this. And you can see the problem with 3 colors is that although you can see individual astrocytes as blue, yellow, or red, this is probably the size of an astrocyte. This is almost certainly many yellow cells, and this red thing is clearly not 1 cell, but many, many cells. And all these black objects in here that are invisible, well, those are the nerve cells, the neurons of the brain, which in this particular transgenic animal were not expressed at all. It turned out, though, that when we made these animals, often this construct was put in tandem multiple times into the same animal, into the same chromosome region, and each construct independently would end up being red, green, or blue. So you might have 1 cell with 3 doses of red, a dose of green, and a dose of blue, and that cell might be brown. Another cell might have just randomly a dose of blue, red, and 3 greens. It might be spearmint color. And then in a line of mice where the nerve cells are expressing, this is the hippocampus and this is the cortex, now one can see lots of colors. And if I zoom up on the dentate gyrus of the hippocampus, you see there's a lot more than 3 colors here. But importantly, although there's a lot of colors, this is still just 3 images, a blue image, a green image, and a red image superimposed to generate all these colors. And we've made new generations of these mice. We call these mice rainbow mice, where now the colors are brighter. And I won't go through the technical details of how these mice are made and how these new generations are better, except to say that we now have very bright mice, and this allows us to think of these wires that connect nerve cells from one place to another as labeled. And this is a brilliant idea, but we did not invent it. It was invented by computer scientists when they were trying to understand how wires go from one part of the nervous system of a computer to another part, and they used to use these labeled lines. Well, that's the level of sophistication we are right now. If you look at these lines and where they connect, and with these bright lights, maybe this doesn't do this full justice, but these are the synaptic connections at the terminal regions of each of these wires coming into a muscle. These are the synaptic sites, the neuromuscular junctions. And because only one purple wire comes into this muscle and both of these adjacent neuromuscular junctions are purple, I know even without tracing these axons all the way back to the spinal cord that these two synapses are coming from the same nerve cell body. And I know equally certainly that the adjacent cell here is a different neuron, and that's 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 a different neuron. In this way, one can map out the entire wiring diagram, the connectome, if you will, of a muscle. And for most of you, you think that, well, why would you ever want to do that? But that's where I spend most of my time, is studying things in muscle. And I'll show you an example of one of these muscles. This is the interscutularis, a little muscle that wiggles the ears of a mouse, pulls the ear actually towards the midline. Each of these is one of those neuromuscular junctions that I was rotating in the previous picture. And Julu painstakingly mapped out every single wire. There are 15 axons coming in, and they contact these muscle fibers running invisibly up and down like this in this muscle, which is a three-dimensional structure, all flattened down here. And this was about 600 gigabytes of data for 15 axons wiring diagram. So it's a lot of data, and it was a lot of computer-assisted human segmentation. That's just a joke. And here is the result. That's the wiring diagram of this muscle. And it immediately allowed us to ask questions like, does this wiring diagram look the same from one animal to the next? Does it look mirror symmetrical on the left and right side of the same animal with the same genes, even the same experiences? And the things we found were somewhat surprising to many people. The first thing was that this wiring diagram, despite the fact that the nerve comes into the same place on each side of the muscle, on the left and right side, they are mirror symmetrical up to this point. Once these axons get into the muscle, every instantiation of this wiring diagram is completely unique, not just between one animal and another, but even between the left and right side of the same animal. Every left side of one animal is different from every left side of another animal, and every one of these wiring diagrams is unique. This is true in mammals. It's not so true in invertebrate animals like insects, and much more highly evolved animals, if you will, that have been around a long time and have perfected their wiring diagrams based on genetic information to do a particular task. In the mammal, we're much more rough, and every animal kind of ends up with its own unique pattern. Not only is it unique, but it's in many ways under-engineered. There's lots of suboptimal wiring, useless loops, premature branching, things you don't see in more advanced animals like insects and worms. And I know you think, well, you don't mean more advanced. I do mean more advanced. Those animals have been around longer. They've evolved more. They really have perfect nervous systems. They don't have to learn anything. They know how to use their brains without going to school. We take a year to figure out how to walk, 18 years, God willing, to leave the nest. I mean, it's just a different thing with us. A lot of this is because this information that's embedded into these other animals is just not embedded in us. We have to bring it in from the outside. That's our great strength, but it's also our great liability. We're helpless for much longer. So anyway, here's an example of a useless loop. This was seen in one animal, but things like this were seen in every animal. Here is one axon. I'm going to just zoom in on this area where all the axons go this way. This red axon has decided, however, to go this way and not go that way. It's the only axon that only goes this way. There's another axon that branches and goes both ways. This axon goes all the way up here and then makes a hairpin turn, comes all the way back down here and then makes a hairpin turn and goes out there. And this branch here is especially interesting. There's a synapse from this axon. This would have been the smart way to get there. This is more the way I drive, my wife says, is that I want to go here, but I remember there is a way I know how to go, so I take this insane route, and I end up there, but I could have saved 8 miles by just taking the shortcut if I just could mentally see that. So there's one example. This neuromuscular junction here is due to a branch at this point. So it would have made perfect sense to make that branch and go this way, but no, it makes the branch there, then follows all the way here, then defasciculates, crosses over itself, and makes that synapse there. These kinds of things suggest a nervous system that doesn't know exactly how to wire itself up, but works out a result that works out fine. And I won't go into the details about this, but I just want to tell you that this wiring diagram at birth is completely different than it is later on. That is, in mammals, this wiring diagram evolves over the course of a lifetime And in a mouse that's only the first couple of weeks of life, it settles out into a wiring diagram by changing. And the change is virtually all loss of wires. At baby animals, at a time they can't use their motor systems very well, have far more branches than they have later on. And what you're seeing here is the culling away of all but a small subset of the branches that were present originally. I won't go through the details of this just for time reasons, but just to show you that whereas in the adult, an axon might innervate maybe 5 or 10% of the muscle fibers in a muscle. Each of these gray sites is a potential synaptic site. Here's one axon, and this is the sparse labeling of the sites it ends up with. If you look at the same kind of axon at 1 day after birth, you find that it innervates 80, sometimes 90% of the muscle fibers. Each axon has about 10 times more branches at birth than it does 2 weeks later in a mouse, and this is probably also true in human beings. And this implies if each axon is branching to many more targets, if the number of targets is constant, which it is, that there must be many different axons converging on each of these sites. And electron microscopy allows us to count the number of different axons coming into one site at young ages. And I'll just show you an example of this technique that I'm going to spend a little bit of time talking about, this electron microscopy technique, where we serially reconstruct a block of tissue. This is a postnatal day 0 mouse, and color in the muscle fiber in green and the axons converging on that. Unlike that pretzel-shaped single axon that innervates each neuromuscular junction in the adult, you can see in babies there's clearly more than one axon coming into that muscle fiber. This is a single muscle fiber, and they seem to be in an orgy of happiness. I mean, it looks like they're going to be there a long time, but in fact, all but one are going to be gone within the next 2 weeks. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and one that's already being eliminated. So this would imply that the brains of mammals, at least in the nervous system and muscle connections, start out with a lot more connections than we end up with. A kind of scary way to think about this is that you start out with a nervous system perhaps wired up for any one of an infinite number of possible things you could become, and then through the efforts of your parents, your teachers, television, the vast majority of those things get pruned away, and you're left being the narrow-minded adults, at least the way my daughter thinks of me, that we become. It's a painful, so education is destructive in a way. It rules out things, and it may be why adults have a hard time agreeing on things, because their worlds may be sculpted by different experiences in childhood, whereas children can probably get along a lot better. Anyway, so 90% of the branches are eliminated. So much of our work since what I've just shown you has been trying to understand what determines which connection stays and which goes. And this is required to do connectomics now in neonatal animals rather than in adults. And we use the same technique, but now instead of neuromuscular junctions that have one input, we start at a week of age where many of them still have more than one axon converging. And from that, we can generate through looking at each of them in the serial imaging reconstructions, we can find the name of every axon at every neuromuscular junction in a baby animal. And when Ryan Draft, who was a graduate student when he started this work, began doing this, he said to me, "What am I looking for?" And I told him, "I don't know. All I know is that imaging has the power to show you things that may go beyond what you imagine. And so if we just look, maybe we'll see something interesting." He said, "Well, what?" I said, "If I knew, there'd be no point in doing this. That's the point of doing this." And we had an argument a long time about this. But finally, after much effort, he came up with some maps. And here is an example of what a map of connectivity looks like in a baby muscle. This is a muscle innervated by 6 axons. It's a small muscle in the neck. And each of these colors is a different nerve cell innervating muscle fibers. And what he did, so this is the red neuron and this is the blue neuron, is he counted the number of muscle fibers that shared inputs in this developmental age at postnatal day 6 between various neurons. And he found there were 66 muscle fibers that shared innervation by those two neurons. That was the most common thing he saw. Then the next most common was 61 muscle fibers sharing those. 60 muscle fibers shared input from those two, 54 from those two, 50 from those two. So, you know, if you had to hazard a guess, what would you guess would be the number of connections between those two? 1. So that was not the next most common connection. The next most common was 29 between those two, which in this particular order skipped 1. 22 between those two. 21 between those two. Each of these are skipping 1 in this particular order. 18 muscle fibers shared connections from those two. 16 from those two, that skips 2 in this order. 7 from those two, that skips 2. 6 between those two in this order, skipping 2. 4 between those two, skipping 3. And there's that 1. So something weird. What is this? What is this? And it took, I'm sure some of you immediately see this as a perfectly obvious pattern. It took us a year of looking at this and doing many muscles. Every muscle had the same kind of weird pattern. Some very strong connections, some very weak connections. What finally made it clear to us was just taking only one neuron at a time. You look at this red neuron, it's very closely associated with this neuron, 66, less so with this, less so with this, less so with this, and least with this neuron. And I've drawn this as a circle, but that's a mistake. If you want to use your visual system, you should graph this in terms of proximity. These are closely associated and these are distantly associated. So we just changed the way we map this and mapped it this way. So for this neuron, it's very closely associated with this neuron where there's 66 muscle fibers that transiently are co-innervated by these two, all the way down to only 1 connection between these two. And we did the same for this neuron. And you notice when I do this neuron, I'm not changing the order of the colors down here. Its nearest neighbors were its strongest associates. Next so here, and no connections there. This neuron, the same pattern applied, and the same pattern here as well. The strongest connections with the nearest neighbors, weaker connections 1 away, weaker 1 away still. Nearest one strongest, weaker, weaker, and here is the last neuron. And in fact, except for this one teeny error, basically, from this pattern, this neuron makes strong connections here, weaker here, no connections here, but 1 connection here. That certainly could be within the noise of our measurement. All of these neurons obeyed some rule, which is there's this linear order where they all work out together. But what is this? We were not looking for this. We had no idea what this meant. But then when we looked at every muscle, they all showed basically the same pattern. I talked to some mathematician friends of mine at APL, at Johns Hopkins, who told me that this is a one-dimensional, latent variable, random bipart graph model. And that didn't help me at all, because I'm not a mathematician. But I did see that this is, rather than a circle or a branching tree, there's an order. There's an order. There's 2 ends and 1's in the middle. It would be as if these were neighbors on a block, on a street, and you ask, how many words do you talk to each neighbor in a week? Your nearest neighbor, you might say 66 words, your next nearest neighbor 29 words, and you might have just a grunt for the person at the opposite end of the block. And these guys at the ends have no one on either side of them, so they're the ends of a block. So this is some order. So that's what we thought. This is some kind of linear order connectional matrix. And I'm just going to make a long story short. That linear order connectional matrix turns out to be the order in which nerve cells are being recruited in development. This is the neuron that is firing first when a muscle is used. This is the neuron firing next, 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 and next. This is what we're working on, the physiology of this right now. What this means is that neurons that fire in a fixed order are connected so that neurons whose firing pattern is most similar to the neuron that's firing before them is most connected with them and less connected to neurons that are firing farther away. That this is actually a map of the order of firing, and therefore this neonatal wiring diagram structure, just the physical structure per se, reveals the experience that set it up. Neurons that fire together remain connected together longer than neurons that don't fire together. So just now, once you've decoded this map, just looking at the wiring diagram, it reveals the experience that set this up. And that, of course, is in grammatization. This is an example in muscle where one just by looking at the wiring diagram can understand now, without having to do the activity patterns. Once we finish this work, we believe just looking at the wiring diagrams will allow us to understand the experience that set it up. But what about the brain? To make a long story short, we are in big trouble in the brain because although these colors are fine, when you zoom up and look, there's just, surprise, way too many wires in between these nerve cells. And so optical techniques don't work. We've come up with some very snazzy new viruses. I'm not going to talk about them in detail except to impress you with how complicated they are. But those viruses allow us for subsets of neurons in the brain to get every neuron a different color. When we're dealing with sparse labeling, we can see, for example, all the inhibitory neurons in a region and see each in a separate color. This is going to be very powerful work. We're doing this with Takao Hensch's lab, which is interested in inhibition and development. But for the central nervous system, we've gone to another approach. You're going to hear in the course of this meeting about yet another approach that is one that uses block face imaging that Moritz will be talking about tomorrow. This is an approach where one takes brain tissue and cuts it extremely thin with an automatic tape collecting ultramicrotome, and that's basically what is shown here. It's a block of tissue that is being cut with a diamond knife floating on water, and then a conveyor belt of tape is taking up section after section after section. And these sections are therefore contiguous regions of the brain, one after another. We cut around 30 nanometers, which is pretty thin for cutting thin sections. And these sections are very big. They're several millimeters on each side. And then we dry them. So we've linearized the brain, basically, and then we cut the brain up into segments. We put them on silicon wafers to image them with scanning electron microscopy. Bobby Kasturi is the person who realized that probably the easiest way to image these would be with scanning electron microscopy. The person who developed the cutting technique was Ken Hayworth, who's now at Genelia Farm. But when he built this with Richard Schleck, we went through several iterations in the lab, and finally the process we have now can cut over 1,000 sections. Much value being a single section. You want to see this in section after section after section after section. Here is just a low resolution rendering of many sections in a row. This data was taken at 3 nanometers per pixel, but I can't show you this data at this size, at 3 nanometers per pixel, because I need 100-fold more area than I've got here. But you can see nerve cells, and these things running around, of course, are just sections. You're all imagers, so you appreciate that nothing's really moving here. This is just going through a data set. Get a better sense of what's actually going on here. We have to zoom to higher resolution. And if you do that, you see that there are vesicle-filled profiles everywhere, these things with little circles in them. Those are the synapses. And these things running around are all the wires of the brain. And, you just pick one of them. You can follow it with your eye. Humans have no problem at all following these. Any child, if you give them this as a coloring book, could color this stuff in. We are trying to make computers as good as children coloring in. But Daniel Berger and Sebastian Sung's lab developed an algorithm that makes a digital coloring book kind of fun to use. And in this way, each of these objects is colored in by a human being, in this case Daniel himself, and then each image is shown one after another. So this is a segmented data set. It took him 2 months to do 432 cubic microns. That's not much, and that's a long time, and that's only 6 by 6 by 12 microns. But if you do that, you then end up with, this is the same data, you just turn it back into 3-dimensional objects. Those are the dendritic spines. And then we put in the axons. So this is totally useless data, absolutely, because every side of this box is orphaned. There's no connections to anything. And making matters worse, that box is there in this image, and that image is there in this section that Bobby was holding for you. So what we were doing is smaller than 1 pixel there. So we're not, and we were taking that at 1 million pixels per second. At that rate, to do a cubic millimeter, a square millimeter is about this big. to do a cubic millimeter of brain at the rate, not the tracing part, just taking the pictures to get to the point where you can start coloring would take 140 years. So we have to go faster. We've come up with ways to get it up to 40 million pixels per second using backscattered electrons and better detectors. And then more recently, we've been working with Zeiss, who's made a 61-beam prototype scanning electron microscope, which gives us another factor of 60 or so. We're hoping to get to 1 billion pixels per second in a couple of years. This is a big machine by scale to a normal-sized human being. Naked, it's got 61 electron microscopes basically running in parallel. So this is an example where the computer technology is an essential part of this feature. So eventually we should be able to do what was taking 140 years in a couple of days. But that's just taking the pictures. One still has to come up with a way of segmenting this area that was done in 2 months. One wants to be able to do unlimited areas. And this is an example of an algorithm that was developed by Hans-Peter Pfister's group at the School of Engineering at Harvard with Verena and Emilio's help. They worked 5 years on this algorithm, and it's a very good algorithm. So this is now 27,000 cubic microns, and after 5 years of setting it up, it took 14 hours to do this. So this is about 6,800 times faster than Daniel Berger. So that it's better, and at this level it looks pretty good. The Golgi apparatus and nerve cells are a little psychedelic. The algorithm doesn't know anything about Golgi. We don't really care about Golgi. But if you look at most of the processes, you can follow them and they keep their color. They look pretty good. If you zoom up at high resolution, this again is low resolution, you see, however, we still have work to do. Although it segments perfectly the right objects, every time an object changes color, that means the algorithm got confused. These are not insurmountable problems. In fact, I already have a rendering that's 5 times more accurate than this one. So we think we're going in the right direction, that eventually we'll have automated algorithms. I just want to end by giving you a sense of what one can do. One can now take a piece of brain, segment all the nerve cells, and focus in, for example, on all the synapses made on a couple of nerve cells that are right next to each other. This is work Babi Kasturi has been doing. Here are all the axons in a region that are innervating these 3 dendrites. There's another 10-fold more axons that have also been traced that run right through this area that don't innervate, don't contact these cells. These are the ones that do. And from that, it's possible to see some remarkably strong connectivity, nerve cells that make many connections on a single cell. So this is sort of the state of the art. For us at least, one of the questions might be are the synapses and axons making on the dendritic spines, are those synapses special in some way? And one way to do that would be to count the number of synaptic vesicles in every single synapse of an axon, since we can see those. That's a lot of work, but I work in an undergraduate campus with a lot of people who want to go to medical school, and they'll do anything for a letter of recommendation. And so here is not an artist's rendering, this is actual letters of recommendations, all shown here. These are the synaptic vesicles and synapses on the dendritic spines of one of these cells. We're hoping, not only will this show us something about how the numbers of synaptic vesicles vary from site to site. Here's one other picture of this. But also perhaps in diseases like autism, it's likely that there are going to be, or schizophrenia, there's likely there are going to be problems at the level of subtleties like this. So this is our hope for the future. So I'd like to just acknowledge the fact that I didn't do any of this. This was done, the photon work, especially by Ryan Draft, and the electron work by Babi Kasturi and Ken Hayworth, especially Daniel Berger's help. And I have a lot of collaborators from engineering and computer science, Hans-Peter Pfister. Josh Sains was very essential for the Brainbow work. Sebastian Sung at MIT, we're doing a lot of work on segmentation. Art Wetzel, right here, and his colleagues are we're dumping a lot of data to them starting yesterday, and we're hoping they're going to help us with many aspects of this. And then at APL, Josh Vogelstein and Paul. And I have support from a number of sources. So thank you very much. I hope I eliminated some synapses today.
a couple of questions.
Beautiful talk. Have you been thinking about the rules
Yes.
that underlie the recruiting?
Yes.
There's more general rules than, if I don't fire together, I disconnect.
Yeah, so that would seem, that's such a common rule. Fire together, wire together, out of sync, lose your link. And that's an idea that's been around forever. My sense is that mammals have not had time to evolve thousands of learning rules, and that the nervous system, the cells in one part of the nervous system, don't know, oh, I'm the motor system or I'm the visual system. They're just nerve cells. So my view is that probably Occam's razor might have some value here. There maybe are a few rules that are used over and over again, and that what makes the visual system different from the auditory system, different from the motor system, is the firing patterns, which are all related to the sensations that come in through hair cells in the ear or retinal cells, which give each system its own peculiarities related to firing. But maybe once the information gets into the brain, basically the same rules apply. So, yeah.
So, in the same way that you built this up from the images, have people taken a parallel approach to the cellular automaton type people and then try to take those rules and say, can I generate these wiring patterns with simple rules?
Yeah, I mean, this is one of these really philosophically dangerous and complicated things. We're first of all using learning algorithms to teach computers how to decipher these images, which are themselves learning algorithms of how brains decipher information. And at the moment, this is just sort of funny to say, but I think eventually this is going to be a deeply problematic circularity in here, that we are limited by our understanding of those learning rules to teach computers. And the only way we're going to get that information is by looking at the wiring diagrams, and the only way we're going to get the wiring diagrams is using computers. So, I think that's really going to happen. It's going to be a weird, interesting time. Maybe that's the singularity we're coming up to.
Alan, maybe last question.
What about what do you think is the future of real-time connectomics, where people use optical probes to perhaps figure out these firing patterns and be able to overlay that on the wiring diagrams that you're delivering?
Yeah, it's essential. I mean, don't mistake this for an explanation of the brain's function. Just as genomics is a map of the atomic structure of DNA, it's not a dynamic map of when particular genes turn on and turn off. This is not that either. This is just a base. It constrains theories. One has to use imaging technologies that allow one to assay activity. The question is how to do that in living animals at the resolution level to see single cells. And Moritz may talk, in retina this is already doable. That is, one can look at particular cells in the retina whose firing patterns one knows, and then look at the EM reconstructions of what those cells and how they're connected. In the cortex, people can do this in certain layers of the cortex better than others, but that is definitely a direction people are going. It both have to happen, dynamics as well as structure.
Thanks again, Jeff.